All right, well, here is our lecture for Wednesday. And so, as you listen to this, be prepared on Wednesday to address some of these more intricate arguments against abiogenesis and actually deal with these in our discussion. So, is abiogenesis realistic? And so, what you find is that at the time that Darwin published on the origin of species, all the way up until getting into the 1930s, 1940s, even after the neo-Darwinian synthesis in the 1930s, the idea was that cells were very, very simple entities, that cells were basically just a bag of protoplasm with a nucleus. And so, because of that, the idea of life originating from non life seemed like a fairly simple problem to overcome. The thing that we have with molecular biology now is the more we understand about how the cell works, what's inside the cell, the machinery that facilitate the reactions that must take place inside the cell, the more we learn, the more we realize that actually generating a living cell from non-living material is an incredibly difficult problem. And so, you could attempt to make an argument that maybe the original living cells were even simpler. This is an animal cell, and obviously the proposal by most scientists is that the original cells were prokaryotic cells, which would have less sophisticated machinery than eukaryotic cells, at least in some applications, and would have less structures inside of the cell than eukaryotic cells. And if the original cells were even simpler still, you could potentially make this problem less of an issue, although there are still certain machinery that have to be in place, and we'll talk through those as we go through this lecture. So you have in the 1920s, Haldane and another scientist, can't think of his name right now, propose a chemical solution or a chemical explanation for the origin of life, focusing on a reducing atmosphere. And what we mean by a reducing atmosphere is one that is going to actually facilitate electrons being transferred to materials, and one that is deficient or completely devoid of oxygen. And so Miller and Urey took this idea of a reducing atmosphere. You can see that represented here with water, methane, ammonia, and hydrogen gas, putting into a chamber, provided some energy in the form of an electric shock. And when you present this atmosphere with that amount of energy, what you actually are able to produce are several amino acids. So beyond Miller and Urey's work, other people have demonstrated the ability to actually make DNA and RNA nucleotides, demonstrated the ability to make about 14 out of the 20 amino acids using a reducing atmosphere. Again, making sure it's does not have any oxygen present, and providing an energy source. Now, this presents an interesting solution, an interesting potential solution to the problem of generating living cells from non-living material. If you could take a gaseous atmosphere, supply the energy necessary to facilitate some chemical reactions and actually generate amino acids and nucleic acids, then you at least could generate the materials necessary to build living cells. There's some interesting problems with this, some of which I'll point out now, others we'll deal with on Wednesday. But yeah, the first one being that In addition to the amino acids that you generate and the nucleic acids, you generate a lot of just organic sludge that actually prohibits many of these amino acids from polymerizing, which is going to be an issue. If you can't polymerize amino acids, you can't make proteins. If you can't polymerize nucleotides, you can't make DNA and RNA. Additionally, you only make the simplest amino acids like glycine in large numbers. The rest of them are made in very, very small numbers, providing a very dilute solution of materials to actually build complex proteins. Probably the biggest problem is that there's very little evidence to suggest that there ever was a reducing atmosphere on Earth. So let's deal with that. Is a reducing atmosphere realistic? And so the simple answer to that is no, and here are some of the reasons why. UV light leads to the decomposition of water, and the decomposition of water generates hydrogen gas and oxygen gas. And so as long as sunlight is hitting water vapor in the atmosphere, it's going to lead to the decomposition of water and the generation of oxygen. So if there's water in the atmosphere, very quickly there's going to be oxygen in the atmosphere. On top of that, Precambrian rocks, those beneath what we would consider to be flood deposits, and some of the earliest rocks that we have on Earth, demonstrate that there was free oxygen present, maybe even in numbers similar to what we have today. And so on top of this, even if you could generate all of the amino acids necessary, which so far we've only been able to demonstrate 14 of 20, and you can demonstrate all of the different nucleotides necessary, we also have an issue that we just have way too little material to fully polymerize these amino acids and these nucleotides and generate proteins. And so there's a calculation provided in the text that demonstrates that we would need something like 1 to the 100 and something universes worth of water just to make a single molecule, make a single protein molecule 100 amino acids long. And so what we see is that even if you could generate all of the materials necessary, these reactions couldn't take place in water because the solutions are too dilute. And so you have a couple of attempts to deal with this. We've got one by Sidney Fox over here on the left. He carried out most of his work in test tubes, but what he did is he provided a pure collection of amino acids, the proper isomer for amino acids in living forms, and he melted amino acids and actually used that fluid to act as the solvent for the other amino acids. And so what he created was these proteinoids, which he equated to being cells, in which you have these collections of amino acids polymerizing inside of a solution of amino acids that had been melted. And he demonstrated that this could take place outside of a test tube, say on the surface of a lava rock. Additionally, you find people that have done work with clays to demonstrate that the porous nature of clays and the really complex structure of clays draws materials in, including amino acids, separating them from the water outside of those clays and giving them an environment free of water in which they could polymerize. And so both of these are presented as an explanation for how we could deal with an extremely dilute concentration of amino acids, get it outside of water, and you could start to concentrate these amino acids. The problem with the clay is what you end up realizing is you can't start with pure amino acids. You actually have to start with a chemically modified version of all of the amino acids in order to get them to polymerize into the form that you need. And the problem with this over here is when you provide pure amino acid of the right type, you actually get polymers of all different types of amino acids with all different types of bonds between the amino acids. You don't get the kind of polymerization that you find as amino acids are strung together to produce proteins. Well, another alternative is maybe we don't start, maybe the original cell doesn't start with proteins, so we don't necessarily have this issue of how do we string together these amino acids to build these complex proteins initially. Maybe what we start with is RNA. It's called an RNA world. And so we know that RNA can be catalytic. We've seen that in ribosomes, where you've got a ribozyme that actually catalyzes the addition of a new amino acid attached to the tRNA that's coming into the ribosome, facilitating it's called amino acid transferase, facilitating the addition of that amino acid to the growing polypeptide chain. So we know RNAs can be catalytic. On top of that, we kind of have these ideas, well, we know RNAs can also store information, and so we have this idea that RNAs could be both the genotype and the phenotype for this organism, and that they're storing the information, but they are also actually carrying out the work of the cell, expressing the information. I have a couple of issues here. First of all, we've never found any RNAs that are self-replicating. This is an additional issue that wasn't pointed out in the text, but RNAs are incredibly unstable. So DNA is a fairly stable molecule, but RNA is actually incredibly instable or unstable. On top of that, we have an issue of how do we, even if that were true, that RNA is acting as both genotype and phenotype, by what mechanism do you get from there to our current central dogma, where you have DNA is storing the information, gets transcribed into RNA, and then that RNA gets modified, matured, and then gets translated into protein. So by what mechanism do we actually get that? So this and other attempts to demonstrate how abiogenesis might work has inspired. This is written by two evolutionary biologists, J. Brooks and G. Shaw. These experiments claim abiotic synthesis for what has in fact been produced and designed by highly intelligent and very much biotic man. Some nice, humility and honesty when it comes to trying to attempt to explain how abiogenesis might work. So now let's for a minute assume that we could actually generate all of the 20 amino acids needed to make proteins, assuming we could generate all of the nucleotides necessary to make DNA and RNA, and we could get it in the high enough concentration to actually polymerize these to build proteins and to build nucleic acids. And assuming all of that can happen, we still have another problem, maybe even bigger than the two previous problems, in that what we're building is not something that just has order. So here you have a crystal, highly ordered structure, and here ordered carbon atoms built into this nice sophisticated crystal. But living entities don't just have order, they have what's called specified complexity. That it's not just a matter of packing in the materials into a crystal, but they actually need to contain information. They need to be strung together in a very, very specific way. The text actually does, I think, a really good job of demonstrating how specified complexity works, showing you letters strung together in order to represent a very specific sentence, or something much more complex than that, like a living cell. You have to string together these these molecules in a very specific order in order to convey that information also forces you to really ask the question of what is the source of that information. So how important is the structure really? So, we mentioned the specified complexity. Do these amino acids really have to be strung together in a particular way? And the text actually talks about two examples of highly sophisticated proteins. Here we have ATP synthase, a protein that takes ADP, attaches an inorganic phosphate to it to generate ATP. I'll call it ATP synthase or the ATP pump. And so as hydrogen ions flow from the inner, the space in between the two mitochondrial membranes into the matrix of the membrane, it's forced to move through this pump. As it does, it turns the head of this ATP synthase and provides the energy necessary to force that inorganic phosphate onto the ADP to create that bond and generate ATP. Highly sophisticated protein, certainly not the most sophisticated protein that you find in cells, but a highly sophisticated protein that requires incredibly accurate construction and has to be built in a very specific way and requires the amino acids being strung together in a very specific way containing the right information. On top of that, the instructions needed to transcribe into RNA so that RNA can be translated into protein require that the nucleotides be strung together in a very specific and the correct sequence. Kinesin, another very sophisticated protein, sometimes called the donkey protein, walks along a microtubule. Every time it walks along the microtubule, it requires a molecule of ATP made through ATP synthase. And so these two proteins are related very intimately, but both are great representations of the sophistication necessary in protein design. So is biogenesis reasonable? I think you'd have to be honest to say the answer to this it seems to be no, but maybe if we're given enough time. And so this is an old illustration that basically if you give chimpanzees a typewriter and you give them millions of years of time, the probability of them typing out one of Shakespeare's plays is incredibly low. But if you're given a number of chimpanzees with a number of typewriters, and again, millions of years of time, even though the probability of this happening is very low, you have so much opportunity for it to happen that eventually it's going to happen. And I think Brand and Chadwick do a good job of taking this illustration and saying, well, yeah, that may be true if, in fact, every time they typed a page, you could make sure those pages stay together, and you could make sure that the typewriters remain, well cared for and well kept for that millions of years of time, and that they kept, they had sufficient materials necessary to type that. Then yeah, maybe, but that's assuming a lot, and they actually make the argument that even given enough time, it doesn't really matter how much time you give them. These chimpanzees are never going to be able to produce one of the works of Shakespeare. Well, maybe this may have happened on another planet. We know the challenges associated with a really dilute concentration. We've got a planet that's predominantly water. And so, well, maybe abiogenesis happened somewhere else. And it's an interesting idea, especially with all the challenges that we have trying to generate the original living cells on Earth. But all it does is move that problem to another planet somewhere else. So is it even possible to overcome these barriers? Can we reasonably suggest not that abiogenesis is true, but really that it might be maybe possible? So even if we were able to assemble everything needed in the cell, we were able to get all of the amino acids we needed, We were able to polymerize them into proteins. We were able to get all the nucleotides we needed. We were able to polymerize them into DNA and RNA. We would still, every reaction that takes place in a cell, in a non-living cell, is at equilibrium. So even if you are able to get all of these materials into this protocell, you're going to need some way in which you can jumpstart these reactions, and you can actually start to take reactants and produce product. That's a big problem of what is actually going to jumpstart a cell. And you may not have thought of that as a problem, but if you think about it, and you think about a cell that has died and is at equilibrium, to actually get that cell to come back to life is an incredibly difficult process. So on top of that, the second law of thermodynamics often uses an argument against evolution, against abiogenesis. And, the second law of thermodynamics doesn't state that you can't increase order of a system. It just says that if you're going to increase order, that you have to be able to input energy. But it's a little bit more complicated than that. So the second law of thermodynamics really requires the following if you are going to be able to generate a cell from a collection of amino acids, even a collection of proteins and nucleic acids, is you need an open system. So you need a way in which that cell can get materials and energy from the universe. On top of that, you need an adequate amount of energy to actually to facilitate or generate a more ordered system. On top of that, you need a machine to utilize that energy. So you need sufficient energy, adequate energy, but you need a machine to be able to utilize that. And the illustration Brandon Chadwick give in the book is, if you've got two rooms that are completely messy, in order to take a room and to make it more ordered, you have to input energy, but you need a machine, such as an organized teenager, to actually take that energy, utilize it to generate that order. On top of a machine to utilize energy, you need an additional machine to control that utilization, to ensure that nothing is wasted and that the energy that is being utilized is also being stored up for when other reactions may need to happen at another time. So is it possible to overcome these barriers? Likely not. It seems very difficult to try to overcome these issues. So an important point to make is, just as believing in interventionism requires faith, right? You have to have faith that what the Bible teaches about God's role in creation is actually true. Abiogenesis requires faith, and it requires an enormous amount of faith to believe, as Richard Dawkins says, that the process of abiogenesis could not be directed by natural selection, because in order for it to be directed by natural selection, you need to have cells reproducing themselves. But what you have is that basically what Dawkins says is just complete random chance to generate the first living cell from a collection of organic materials. And even to generate the first organic materials from inorganic material requires just pretty much dumb luck and chance, because there was no outside force to drive that process. And so this requires an enormous amount of faith and really a commitment to methodological naturalism. But methodological naturalism requires abiogenesis, requires life generating from non-life. And so while having a God or a creator that's separate from his creation and immaterial, speaking life into existence, may be easier to believe than abiogenesis, a commitment to methodological naturalism really requires abiogenesis. So as you listen to this, remember that in Wednesday we're going to talk through some of these arguments more specifically. We'll deal with them and hopefully we'll get you a good sense of why it's very, very difficult to support a view of abiogenesis.